Alright, so we're doing E. coli lactose metabolism in prokaryotic cells. So we have our portion of DNA here. See, they have the loop DNA, and this is the operon portion that we're dealing with that goes with lactose. And so we have in dark blue the repressor, which helps in regulating the production of proteins that metabolize lactose. We have RNA polymerase that's going to move down and make RNA, mRNA out of the genes we have. We have the pink as the portions of the chromosome that we're not looking at. Out here we have lactose. That's what's being metabolized. In light blue clay, we have Lac I, a gene that produces repressors. In red, we have Lac Z, the gene that produces proteins that break down lactose molecules. In orange, we have permease, which helps lactose molecules access the cell easily. And then finally, with permease, we have the protein made by Lac A, which detoxifies other molecules that get in through permease that aren't lactose. And here we have ribosomes just within the cytoplasm of the prokaryotic cell that will make proteins out of the mRNA. Alright, so running through the process of this operon, initially proteins aren't being made because RNA polymerase cannot access the genes because it attaches to the promoter, but then it can't get past the promoter because the repressor is there. So when high concentrations of lactose are in the cell, we'll get there eventually. Allolactose, which is like a cousin of lactose, will attach to a site on the repressor, and that will inhibit it so that it cannot attach back in here. Okay, so then polymerase can come in and it can start scanning molecules and making mRNA strands from these genes. And then those will enter ribosomes, right? And the ribosomes will start making our different proteins. So we have all our different proteins that are going to help in the metabolism of lactose. So the B-galactidose or whatever.
Beta-galactosidase.
Okay.
Yeah.
So this is gonna grab these lactoses and it's gonna split them into fructose and glucose, and then these can be more easily metabolized. So these are gonna keep going around and metabolizing the lactose molecules, and ones outside the cell are gonna be accessing through permease, right, more easily. So instead of having to get through the membrane, they can just come along. And meanwhile, RNA polymerase is watching the concentration of lactose and it's creating repressors as needed. So as lactose gets metabolized and the concentration lowers, the allolactose is going to detach, and that reshapes the repressor, which can reattach so that lactose concentration stays at a safe level instead of going to super low concentrations of lactose. So then proteins stop being produced in the ribosomes. Oh wow, it doesn't erase. Okay, well. And again, RNA polymerase can't access the genes. So basically, when the repressor is active, the genes can't be expressed because polymerase can't access the operon. And so also lactose won't be being metabolized. So this is an inducible operon because the repressor, well, one is disabled through an inducer, but also it is active and becomes inactivated.